the honor of introducing Dr. Monje today. She is an associate professor of neurology and neurological sciences and the scientific director of the Center for Childhood Brain Tumors at Stanford University. She's received her PhD and MD in neurosciences from Stanford, and then her neurology training at the Partners Program in Boston. She returned to Stanford for clinical fellowship in pediatric neuro-oncology and a postdoctoral fellowship, and her research lab focuses on neuron-glial interactions in health and disease, with a long-standing emphasis on the cellular and molecular mechanisms mediating disruption of neuroplasticity following cancer therapies. So thank you very much for your time today, Dr. Monje.
Thank you so much for having me in this really fantastic symposium. Today I'm going to focus on glial dysregulation in cancer therapy-related cognitive impairment. So as many in this audience know well, healthy cognitive function relies upon healthy neural circuit function. And this is really a collaborative endeavor between multiple cell types. So in order for neurons to communicate with each other appropriately, they have to have appropriate synapses, and astrocytes help to promote synaptogenesis, as we just heard so eloquently from Dr. Hervey Jumper. Microglia are in the healthy brain pruning these synapses to promote appropriate connectivity, and can do that process too much or too little, depending on their state. And in order for dynamics within a circuit to proceed appropriately, we have to have adequate and really very finely tuned myelination to regulate the speed of neural impulse conduction. The myelin comes from oligodendrocytes, which are in turn generated by oligodendrocyte precursor cells. And so these different aspects of neural circuit function, also rely, particularly in the adult brain in certain regions, like the hippocampus, upon the generation of new neurons. And so we know that new neurogenesis in the hippocampus proceeds in both animal models, and most laboratories believe in humans as well, and that this is an important contributor to normal memory function. And so what we've learned over the last couple of decades is that radiation and chemotherapy can impair broadly mechanisms of neuroplasticity. We know that radiation as well as chemotherapy can impair the function of neural stem and precursor cells. This is particularly prominent in the hippocampus, where Jörg Dietrich, who we're going to hear from later this morning, has beautifully demonstrated that many different chemotherapeutic agents can impair the survival of neural precursor cells. In my older work with my PhD advisor, Theo Palmer, in collaboration with John Fike at UCSF, we showed that radiation also very, very severely impairs hippocampal neurogenesis, not just by damaging the precursor cells, but by damaging the environment that is required to support neurogenesis. Multiple groups have shown that cancer therapies like radiation and chemotherapy can alter the plasticity of synapse structure and function. And I'm going to tell you about some work today from my independent laboratory that cancer therapies can impair plasticity of myelin, both myelin homeostasis, but also really important adaptive changes in the myelinated infrastructure that we think supports coordinated network function. So as we've heard, so eloquently earlier this morning, cancer therapy-related cognitive impairment is a syndrome that affects far too many cancer survivors. It's relatively common, and it consists of impaired attention, memory, speed of information processing, and multitasking. We think that this affects a large proportion of cancer survivors to a variable extent, and variables that we think contribute to the incidence and severity of cognitive impairment after cancer therapy include the age at which a person receives cancer therapy, the exact agents and strategies used. Certainly things are more severe when cranial radiation is part of the treatment regimen, and particular chemotherapeutic agents are more prone to cause this syndrome, and with a greater severity, as we've heard about. In many cases, standard neuroimaging is relatively unremarkable, even when symptoms are severe. More advanced neuroimaging technologies like diffusion tensor imaging typically do, though, reveal subtle white matter aberrations. And we know from Jörg Dietrich and others' work that there is an acute vulnerability in the white matter. The oligodendrocyte precursor cells are exquisitely sensitive to radiation and to chemotherapy. Well, we wanted to understand what happens to this population in the long term. And so several years ago, a really talented postdoctoral fellow in my lab, who now has her own independent laboratory at Stanford, Erin Gibson, and I began to examine post-mortem autopsy samples from subjects who had received chemotherapy. And what we found was that there was a consistent depletion of oligodendroglial lineage cells, particularly in the subcortical white matter, with relative preservation of oligodendroglial lineage cells in the cortex. Looking in a little bit more detail, we were able to prospectively collect some samples in which we could save the tissue for more precise, immunohistochemistry and confocal microscopy. And what we find is that in comparison to patients who did not receive multi-agent chemotherapy, and in particular high-dose methotrexate, which is a particularly neurotoxic chemotherapeutic agent prone to cause this kind of cognitive impairment. We find that in comparison to in this case, children who did not receive high-dose methotrexate, that children who did had essentially a dropout of oligodendrocyte precursor cells in the subcortical white matter. But we know from the work of Dwight Burgles and others that oligodendrocyte precursor cells are a population that's very good at repopulating itself. So these OPCs should be, after an initial cytotoxic insult, be repopulating rapidly. Yet we see this persistent decrease in oligodendrocyte precursor cell populations in the long term. So we wanted to understand why the cells were not repopulating after methotrexate chemotherapy. And so to do this, we developed a mouse model of high-dose methotrexate in which we give 3 cycles of methotrexate to juvenile mice between the ages of postnatal day 21 and 35. These are levels that achieve serum and brain concentrations similar to what humans experience after high-dose methotrexate. And then we examined these mice either 1 month or 6 months after this remote exposure to methotrexate. And what we found was that just like in the human tissue, there was a decrease in proliferating OPCs, and that this was really specifically in the subcortical white matter and not in the cortex. So looking more carefully at the oligodendroglial lineage, at OPCs, at intermediate cells in between an OPC and a mature oligodendrocyte, and at mature oligodendrocytes, that there was a decrease in the OPC population, as well as in the mature oligodendrocyte population, with actually an accumulation of cells in this intermediate state. And that this dysregulation of the oligodendroglial lineage persisted for at least 6 months after the last exposure to methotrexate. Together with these oligodendroglial lineage dysregulation and deficits, we see a deficit in myelin ultrastructure by electron microscopy, with the white matter in mice previously treated with methotrexate exhibiting much thinner myelin sheaths, as measured here using G-ratio analyses, and that this thinning of myelin persisted for up to 6 months at least. Together with these histological changes, we're able in this model to document some of the deficits in cognitive function that patients experience after methotrexate and other similar agents. So I'm showing you the behavioral testing results from the novel object recognition test. We saw these deficits in multiple relevant cognitive domains. But just to introduce you to this test, as I'm going to show it to you a couple of times during the talk, it's a relatively straightforward test. We take a mouse and place it in a chamber with 2 identical objects, and then we remove the mouse from that chamber and replace one of the objects with a novel object. We were particularly interested in testing attentional function because that's such an important domain of cognition for patients after cancer therapy. It's such a common domain in which patients experience difficulty in daily function. And so we altered this test so that there was a very short interval in between exposure to the objects and then testing to see if they recognize the novel object. Now, a healthy mouse, when placed into this chamber, will spend more time with the novel object because it will have paid attention to, and in this very short period of 5 minutes, have remembered what it previously saw. And we certainly see that in the vehicle-treated control mice. However, what we find is that both at 1 month and at 6 months, that the mice previously exposed to methotrexate spend equal time with the novel and familiar object, underscoring this deficit in attention and very short-term memory function. And so we wondered whether this dysregulation of the oligodendroglial lineage was due to cell intrinsic effects or to microenvironmental mechanisms that were preventing the oligodendroglial lineage cells from functioning as normal. And so we tested this by taking oligodendrocyte precursor cells from healthy mice and then transplanting them into the environment of the previously methotrexate exposed mouse brain. And so we found that when we put healthy chemotherapy naive oligodendrocyte precursor cells into the brains of mice that had previously been exposed to methotrexate a month earlier, that those cells also exhibited dysregulated differentiation. There was an accelerated differentiation of the oligodendrocyte precursor cell. Now, together with this microenvironmental aberration, we noticed persistent and really prominent microglial activation, but specifically in the white matter. So here's a confocal microscope image of this. You see these activated microglia expressing activation markers like CD68 and upregulating EBA1, and it's really a stark increase, but quite specifically in white matter microglia with equal numbers and equal activation state in cortical cortical microglia. This microglial activation appears to be a state change in the microglia. We see this persist up to at least 6 months. Now, when we look at whether this is an activation state that is direct or indirect, we find that through transcriptomic analysis of the microglia isolated from mice that had previously been exposed to methotrexate, that when we take these microglia out in vivo, they exhibit this really broad activation profile, and that exposing microglia in vitro to methotrexate phenocopies this, it replicates it, indicating that methotrexate is directly activating microglia. Now, together with microglial activation, we were also interested in astrocytes. Ben Barris's lab had shown that microglial activation can induce a neurotoxic state in astrocytes that can then induce neurotoxicity in both neurons and mature oligodendrocytes. And so we wondered whether astrocytes were in a neurotoxic state. And when we look at the transcriptomics from astrocytes isolated from mice that had been previously exposed to methotrexate, we do see that the astrocytes are in this kind of overall activated state. Now, if we expose astrocytes directly to methotrexate, that does not replicate this finding. However, if we expose the astrocytes to the conditioned medium from microglia that were exposed to methotrexate, then we see prominent astrocyte activation, confirming that this direct effect of methotrexate on microglia then consequently activates astrocytes in the microenvironment. And so we wondered if microglia are at the center of this microenvironmental perturbation. If we deplete microglia using a small molecule CSF1R inhibitor, might this help to promote normalization of the oligodendroglial lineage, restoration of myelin, and perhaps rescue of cognitive function? And so after giving mice the 3 doses of methotrexate, we then spent the subsequent month giving the mice this small molecule CSF1R inhibitor. And that achieves about an 80 to 90% depletion of microglia. And what we found excitingly is that this depletion of microglia restores astrocyte reactivity to a more neurotrophic state, and it normalizes oligodendroglial lineage dynamics. Together with that, we see a restoration in myelin ultrastructure. And this rescues the cognitive dysfunction that we had observed. So mice perform as well as their vehicle-treated controls after they receive CSF1R inhibition after methotrexate. And so what we're finding is that there is this dysregulation of really all 3 glial lineages after methotrexate. Just to summarize what I've just told you, methotrexate directly activates microglia, which then results in stimulation of a neurotoxic astrocyte reactivity. And together, these reactive glia dysregulate the oligodendroglial lineage, causing a change in myelin ultrastructure and ultimately contributing to cognitive dysfunction in this model. Now, we know that myelin is important throughout life for, adding new oligodendrocytes is important for myelin homeostasis. But as well, there's our lab and others have demonstrated that myelin is really a very plastic structure, responsive to experience and neuronal activity, and that this plasticity of myelin helps to promote adaptive neural change. We know that one of the important mechanisms that mediates this is activity-dependent BDNF secretion from the neurons, which stimulates the TrkB receptor on oligodendrocyte precursor cells. This is a required component of the neuron-glial interaction that drives this plasticity of myelin. And in preclinical models, we and others are finding that this plasticity of myelin contributes importantly to coordinated network function and healthy cognition. It is a critical component of maintaining appropriate attention and short-term memory function, memory consolidation, and learning. And so we wondered whether the plasticity of myelin might be particularly disrupted after exposure to methotrexate chemotherapy. And so we tested this idea, again, using this high-dose methotrexate model, but this time following methotrexate, what we did is we optogenetically stimulated neuronal activity and then looked to see whether we had the expected proliferative and differentiation response in the oligodendroglial lineage that is normally induced by neuronal activity of cortical projection neurons. And what we found is that in comparison to mice that were treated not with methotrexate, but with vehicle control, in which we see that neuronal activity elicits OPC proliferation, oligodendrogenesis, and then changes in myelin ultrastructure, in mice that had previously received methotrexate, what we found is that there was no proliferative response of oligodendrocyte precursor cells, no additional new oligodendrocytes made in response to activity, and no activity-regulated changes in myelin ultrastructure. So there seems to be a complete blockade of these plasticity mechanisms that are in the white matter after methotrexate. And together with this loss of myelin plasticity, we found that cortical projection neurons stop making BDNF after methotrexate exposure. So performing RNA-seq, performing RNA-scope in the brains of mice that had been previously exposed to methotrexate, we find that in comparison to vehicle-treated controls in which you see these cortical, deep layer cortical projection neurons expressing BDNF mRNA, that after methotrexate, that is lost. If we deplete microglia, we can restore neuronal BDNF expression through mechanisms we're still working hard to understand. And so we wondered in the context of this whether augmenting BDNF to TrkB signaling after methotrexate might be an alternative or additional preclinical therapeutic strategy for cognitive impairment after cancer therapy. And so Frank Longo's lab has developed a small molecule TrkB agonist, and we administered this to mice following exposure to methotrexate. And what we found was that, excitingly, this TrkB agonist normalized myelin ultrastructure in mice previously exposed to methotrexate. And when we test mice in the novel object recognition test, we find that this small molecule TrkB agonist indeed rescues cognition, but does so in a way that depends upon TrkB expression in the oligodendrocyte precursor cell. In other words, we know that BDNF to TrkB signaling broadly influences neuroplasticity, but if we specifically look at the effect that it has on the oligodendroglial lineage, we find that those myelin effects are what is responsible for the cognitive rescue that we see after this therapy. And so to summarize what I've just told you, we believe that loss of this activity-dependent myelin plasticity contributes importantly to chemotherapy-related cognitive impairment. In the healthy brain, neuronal activity results in, particularly in cortical projection neurons, results in release of activity-regulated BDNF that stimulates oligodendrocyte precursor cells through the TrkB receptor. This results in new oligodendrogenesis and changes in myelin ultrastructure, both through de novo myelination as well as myelin remodeling of existing oligodendrocytes. And these myelin changes confer network dynamical changes that are important for coordinated network function and intact cognitive performance. After methotrexate chemotherapy, there's a direct activation of microglia, which through mechanisms we're working to understand disrupts neuronal BDNF signaling, completely blocks myelin plasticity, and contributes important to the cognitive impairment that we observe in at least these preclinical models of high-dose methotrexate. And I want to end with the idea that in numerous studies and through numerous mechanisms, neurotoxic microglial reactivity is really central to cancer therapy-related cognitive impairment. We and others have found that reactive microglia, are disrupting neurogenesis in the hippocampus after radiation. We find that there's this disruption as well of myelination, oligodendroglial lineage dynamics and myelin plasticity for which microglial activation is central. Reactive microglia induce a neurotoxic state of astrocyte so that instead of supporting neuronal function, instead of promoting synaptogenesis, supporting synapses and neuronal health, instead these astrocytes become quite neurotoxic. And many folks, including the Rossi lab at UCSF, have shown that reactive microglia promote aberrant and aberrantly upregulated synaptic pruning, decreasing network connectivity after cancer therapy. And so thinking forward at therapeutic targets to try to mitigate these effects, addressing microglial reactivity, restoring microglia to a neurotrophic state, removing and addressing the microglial inflammation that we think to be central to many of these pathophysiological processes, will be an important and hopefully productive therapeutic strategy. Many people to thank. I want to especially highlight Erin Gibson and Anna Garrity, who led the two projects that I just described in the lab, as well as the rest of my great lab and our really critically important collaborators, and of course our funding sources. So I think we have a few minutes left at the end here for questions.
Yes, Dr. Monje, thank you so much. That was such a lovely presentation. A few questions. So one is you're highlighting these reactive astrocytes in the microglia environment. And how do you envision that, or what has maybe been investigated thus far as it relates to immunotherapy and any impact that immunotherapy on the microenvironment may have in this component of your work?
Yeah, thanks for asking that question. We're actually, so my laboratory also studies CAR T-cell therapy for high-grade gliomas, particularly for diffuse midline gliomas. And we're in fact running a clinical trial now of that. And so I'm deeply interested in how immunotherapy may influence microglial reactivity and ultimately long-term cognition. And so we're actually actively studying that right now. And so it's early to report results, but we've been looking at whether or not there is a similar pathophysiology induced by broad systemic inflammation like the cytokine release that happens with CAR T-cell therapy and with other immunotherapies on white matter microglia in particular, which seem to be this particularly sensitive subpopulation of microglia to inflammatory and toxic insults, and that may be therapeutically modulatable using strategies like CSF1R inhibition. So if you have the symposium again next year, I will report what we're finding, but we're actively working on understanding that.
Perfect. Thank you very much. And then using treatment such as the TrkB1 agonist, do you feel that there's anything as it relates to timing of those interventions as a rescue or maybe more as a neuroprotectant, and if you have thoughts on that?
Yeah, absolutely. I think there are a number of questions left to answer about strategies like either microglial, a temporary period of microglial depletion using something like a CSF1R inhibitor, or a sort of, neurotrophic intervention like a TrkB agonist, or actually, metformin, which was a drug that was brought up earlier by Dr. Weffel, shown by Don Mabbitt and colleagues to promote kind of neuroregeneration after cancer therapy that actually probably is working on microglia as well as on neural precursor cells. The timing of it is really important. I think that we have to think both about the way our interventions might affect the cancer that we're trying to treat, as well as, what the appropriate timing is to mitigate some of these responses. One thing that we have found, and this is unpublished, but we have found that if you deplete microglia, when they come back in this methotrexate model, when they come back a few weeks later, they're actually in a neurotrophic state. So we think that a temporary period of depletion might be therapeutic to sort of reset the microglia, but we don't know what the ideal timing is. We've always done this immediately after exposure to therapy. And for people living with cancer therapy-induced cognitive impairment, a really pressing question is, can we do this much later? Can we reset microglia in through whatever mechanism we find to be the most effective? Can that restore these mechanisms of neuroplasticity even after a long period of time? And so those are ongoing experiments that we're trying our best to answer as well as we can in the lab, and that ultimately will require, clinical studies to really answer.
And one additional question is how generalizable you feel like this mechanism of injury with methotrexate is to other CNS penetrating drugs, temozolomide, for example.
So I think two things. I think that many strategies activate microglia in a neurotoxic way. There may be subtle differences in the way that methotrexate and another chemotherapeutic agent that causes this kind of pathophysiology, influences the state of microglia in other cells. But I think that this is a common mechanism to many, but probably not all interventions. And so I think that these subtle differences between agents need to be explicitly studied. We know that there are some chemotherapeutic agents that are more prone to cause neurotoxicity, and there's likely to be additional mechanisms that perhaps methotrexate doesn't exhibit. And so I think each drug needs to be studied pretty in a pretty focused way. But an emerging common principle seems to be that aberrant microglial activation is central to many forms of cancer therapy-induced cognitive impairment.
Great. Great. Thank you so much.